Hello friends, welcome back to my YouTube channel Doctor's Corner. Today we are going to discuss about apoptosis, a very important topic in general physiology and in general pathology. Okay, before going in detail about this process of apoptosis, example and mechanism of action, I would like to give some background. You know, human body have trillions of cells, okay, trillions of cells. So out of these trillions of cells, daily there will be cell death of around 50 to 70 billions of cells in normal adults, okay, normally. And in children, that is from 8 years to 14 years of age, around 20 to 30 billion cells die daily, okay, that is a normal process. So cell death, basically it is of two types. Okay, cell die by mainly two mechanisms. There are other ways also, but broadly there are mainly two types. One is necrosis and another one is today's topic, apoptosis. Okay, necrosis is only a pathological process. Okay, and apoptosis can be pathological or can be physiological, that is can be occur at the normal conditions also. Okay, so this, if to understand this basic topic of apoptosis, I am giving this background just to understand in a simple way. You consider necrosis as a cell death by a murder. Okay, if you murder someone, okay, so consider murdering of a cell, it is necrosis. Okay, and suicide, cell death due to suicide, okay, that it is considered as the apoptosis. So apoptosis is nothing but it is a type of a programmed cell death in a multicellular organism. To define apoptosis, I repeat, apoptosis is a type of programmed cell death in a multicellular organism. Okay, so it is like a cell suicide, programmed cell death. Cell will be destined to die itself by its own. Okay, it is a neat type of death, whereas necrosis is a murder of a cell. Murder, that is, necrosis occurs only in pathological conditions. Okay, so I hope the difference between this is very much clear for you. There are many differences for that, but I'll just highlight few differences between necrosis and apoptosis because time won't permit me to describe in detail about that thing. So in necrosis, usually while the cell is dying, there is swelling of the cells are seen. Okay, swelling of the cells are seen, but whereas this cell death due to apoptosis, the cell size will be less, that is there will be shrinkage of the cells. Okay, and also there will be chromatin condensation is seen in the apoptosis. Then one of the another important difference is inflammation is present in necrosis, whereas inflammation is absent in apoptosis. Okay, what do you mean by that? That is, when the cells are dying, the surrounding tissue, there is a inflammatory cells are visible or the signs of inflammation are seen in case of necrosis, whereas in apoptosis there are no signs of inflammation. Very clean type of this cell death, that is when the cell is destined to die, it will die its own. Okay, then the surrounding cells, okay, surrounding cells, there will be no inflammation is seen. Okay, then there is a formation of the apoptotic bodies and blebs, and they are engulfed by the macrophages. The details we are going to now discuss under this topic of apoptosis. I hope you have just a gross idea right now what is apoptosis. So it's a time of a programmed cell death. Okay, here the cell will die and their cells are destined to die. So first, before going to the mechanism, let us consider few examples where this apoptosis or which type of the cells die by this process. So it, as I said you, it occurs in both physiological as well as pathological condition. Okay, so physiological condition in which apoptosis occurs, there are many. So I'll just give you a few common physiological condition. One is embryogenesis. Okay, embryogenesis. So during this process of embryogenesis, many cells, they die due to the process of apoptosis. So what exactly you mean by this term? Okay, so you know that human body, a newborn is born, throughout the period of gestation of approximately 9 months, so from a single cell or a 2 cell stage, okay, so it will multiply and finally a newborn is born through the course of the development during the pregnancy of 9 months, okay. So during that process various organs and organ systems are developed. So I give an example. For example, you consider this one 5 fingers, okay. So during the time of development they will be like a bird, and initially there will be no clean cut 5 fingers like this, but as the process of embryogenesis or organogenesis develops, then later what happen, in between these cells will die, okay, in between these cells will die, and ultimately we get this 5 digits and a human hand is formed, okay. So that the cells which die is by this process of apoptosis, okay. Similarly, if we consider a esophagus, okay, esophagus initially it will be just a solid mass of cells, okay, esophagus, okay, but later what happens is the cells in between this will die due to the process of apoptosis, and it will become a tube like structure, and that is an example of apoptosis, okay. So this is an example of embryogenesis. Then another physiological or examples where we see this apoptosis is in hormone dependent cells. So involution or death of the cells which are dependent on hormones, okay. So involution of the hormone dependent cells. So examples in menstrual cycle, okay, hormone dependent cells, or so in menstrual cycle what happen is in females, adult females, so cyclical changes occurs every 28 to 30 days. So the endometrium, that is the inside the uterus, the endometrial cells will shed off, and it is appeared as the menstrual bleeding, okay. That shedding of the cells is dependent on the hormones. So whenever there is a withdrawal of estrogen and progesterone hormones, then these endometrial cells will shed off. So that shedding off, it is due to this is an example of apoptosis, okay. In German it means shedding off of leaves. It's the literal meaning of the apoptosis. Anyway, so in hormone dependent involution of cells, for example, again in lactation, after the period of lactation, okay, the milk producing cells will die, and that is also due to the process of apoptosis. Then many examples are there, again some in proliferating cells, like lymphocytes are proliferating, B lymphocytes and T lymphocytes are proliferating in thymus and bone marrow. So in that also there are some cells will be dying, that in proliferating cells like lymphocytes the cells will be dying, few in proliferating pool. So that is also an example of apoptosis. Then some T lymphocytes, okay, or T cells, they are self-reactive, that is they will damage the own tissues, okay. So destruction of such cells, again an example of the apoptosis, okay. So or T cells, so that is the self cells, destruction of the self cells, this one self antigens. Then again there are so many physiological examples. For example, during the process of inflammation, the neutrophils after its action is over, okay, the neutrophil will die by its own. So after inflammation, dying of the neutrophils is again due to the process of this one. And in immunity, the various cells like lymphocytes, they will die. So that is also the physiological process of apoptosis. Now coming to the pathological examples of apoptosis. So not only in physiological condition, in pathological or some disease conditions also, the cells will die, that is the cell suicide will take place in a programmed way. So that it is known as apoptosis in the pathological conditions, okay. So many a times it can be in your theory university examination for MBBS, it can be a 10 marks or a 5 marks question to describe the apoptosis. You need to give the examples, physiological and pathological examples of apoptosis. It can be asked in general physiology or in general pathology too. Then you have to explain the mechanism of action, which we are going to discuss now. Before that, pathological examples are, see, one of the important thing is DNA damage of a cell due to cytotoxic drugs or irradiation or certain radiations, okay, which will damage the DNA of a cell, okay. So the DNA damage of the cell then there is no repair mechanism. Normally there is certain repair mechanism, but when the repair mechanism is not so efficient and some abnormal cells are multiplying so that there should not be a malignant transformation, then the cells will undergo the process of apoptosis and the cells will die. That is DNA damage. Then abnormal accumulation of proteins, abnormal misfolded proteins, okay, abnormal misfolded proteins accumulation, okay. So at that times also the cells will die, because this may code for different abnormal proteins, okay, that is not normal, okay. For example, in a cell, from this nucleus, DNA transcription and translation process takes place and so many enzymes or proteins are formed. Sometimes due to the misfolding of the proteins, these will be abnormal enzymes or abnormal proteins will be there, okay, that will not lead to the normal functioning of a cell. So sometimes in the endoplasmic reticulum, these misfolded proteins will be accumulated, and that condition is known as endoplasmic reticulum stress or ER stress, okay. At that condition, when there is so many accumulation of the misfolded or abnormal proteins, then the cell will die due to the process of apoptosis. Then viral infected cells, for example, hepatitis infected cells or HIV infected cells, they also undergo the cell death by the apoptosis. Then finally, one more example, see, there will be some stones in the gallbladder or in the renal tissue, that is in parenchymal tissues, where it has a duct, and this duct is sometimes blocked by some stone or something. So due to this blockage, these cells will undergo cell death. That is again an example of the apoptosis due to the renal stones or gallbladder stones, okay, or even the salivary gland ducts, okay. So these are the classical examples of apoptosis. So now coming to the most important concept, mechanism of apoptosis, okay. How exactly this apoptosis process takes place, and how this cell will die, and why there is no much inflammation of the surrounding cells, and how this process is taking place, okay. So we'll discuss now the mechanism of apoptosis. Please give your kind concentration here. If you understand this mechanism, so you need not mug up, you can very easily remember this mechanism of apoptosis. So basically, this apoptosis, that is the programmed cell death, can be taken place by two pathways. One is intrinsic pathway. Intrinsic pathway, it is also known as mitochondrial pathway, okay. Remember, intrinsic pathway also known as mitochondrial pathway. Then one more pathway by which apoptosis takes place, it is known as extrinsic pathway. Extrinsic pathway, that is from extrinsic pathway, also known as death receptor pathway, okay. Remember these two pathways. Intrinsic, that is inside the cell, mitochondria will be there, that is mitochondrial pathway. Then the extrinsic, due to some receptors which are present on the cell, that's why the name given is death receptor pathway, okay. Death receptor pathway, intrinsic pathway and extrinsic pathway, death receptor pathway. So before describing in detail about this intrinsic and extrinsic pathway, I will just tell you the basic thing what happens during the process of apoptosis, okay. Consider this as a cell which is destined to die by the process of apoptosis. So basically what happens is, there is formation of or the production or activation of certain enzymes or proteases that will kill the cellular elements. The cell has nucleus, cytoplasmic organelles and cytoskeleton, that has to be damaged, okay. So for that there will be certain proteases, okay, certain proteases. So consider this as a proteases, okay, that will digest away the cellular organelles, okay. So it will digest the nucleus which is present in the cell, okay. It will break down or it will fragment the nuclear material, then other cellular organelles and cytoskeleton. So this proteases which will digest the thing, and this is the main mechanism for the apoptosis, for the cell death. So these proteases, they are now called as Caspases. So remember this very important enzyme, Caspases. Okay, so basically in intrinsic or extrinsic pathway, the most important enzyme or a protease what we are going to discuss is Caspases. Okay, why the name is Caspases is as the alphabetical you can see with the alphabetical, in this protease there is some active site. This active site, it is there is cysteine molecule will be there. So in the active site of this protease there will be cysteine molecule, and on the it will ultimately digest the nuclear material or the other cellular organelles which are basically protein in nature. So this protein nature, these protein molecules, so on the protein molecules there it is basically made up of amino acids. So there will be one important amino acid, aspartate. Okay, aspartate. So this is caspase molecule or the caspase enzyme or protease, it will digest the different organelles or different proteins where at which site, aspartate. So that's why the name given is cysteine. C stands for cysteine, ASP stands for aspartate, and this is proteases. So cysteine, it is a protease which has cysteine on its active site, and it will act on the aspartate site of the different proteins. Okay, that's why the name given is Caspases. Remember, the most important thing is Caspases. Now these Caspases is generally not in the active state, it will be in the inactive state. Okay, so this active site will be wrapped by something, okay, that will be inactive state. So this inactive state, it is known as procaspases. So inactive state, it is known as procaspases. So when this process of apoptosis will initiate either by the intrinsic pathway or by the extrinsic pathway, so these procaspases are converted into caspases, and ultimately there will be digestion, there will be endonuclease secretion, digestion of the cytoskeleton and other organelles, and all these cells will get digested, and there is a formation of the apoptotic bodies. That is, these caspases will digest this thing, and slowly this cellular, there is a shrinkage of the cell size, and there is a formation of cytoplasmic blobs. So these cytoplasmic blobs ultimately they form certain protein bodies known as apoptotic bodies. Apoptotic bodies. Okay, apoptotic bodies are formed. So this apoptotic bodies is a membrane bond structure in which there will be the fragments of the cellular contents, and sometimes even this nuclear contents will also be there, may or may not be. So this membrane bond structures which contains nuclear material or the other cytoplasmic material are known as apoptotic bodies. So one of the pathological changes that takes place is the apoptotic bodies, formation of the apoptotic bodies, whereas in necrosis we don't see this apoptotic bodies. Okay, that we are going to discuss. So either by the intrinsic pathway or by this extrinsic pathway, ultimately these caspases are activated and they will digest this cellular contents, and there is so many apoptotic bodies are there. Then what is the fate of these apoptotic bodies? Okay, the cell size will shrink, there is a condensation of the nuclear chromatin. So what will happen ultimately with these apoptotic bodies? I'll discuss you. First I will explain you what is intrinsic pathway and what is extrinsic pathway. Okay, hope this is very much clear to you people. That is what is caspases and procaspases. So first, mitochondrial pathway. So remember, in the mitochondria, in the cell, you have one of the very important organelles known as mitochondria. I hope everyone knows this. Okay, powerhouse of a cell. So in this, there is one more very important substance known as cytochrome C will be present within this mitochondrial membrane inside this. So it is cytochrome C. It is present in the mitochondria. Generally it is not present in the cytoplasm, it will be present in the mitochondria, and very much important for the functioning of the mitochondria and for the oxidative phosphorylation and all. Okay, very important molecule, cytochrome C. So generally it is in the inside of the cell. Now how it is remaining inside of the cell? If it comes outside of the inside of the mitochondria, if it comes, the cytochrome C comes outside the cytoplasm, then it will activate this pro-caspases to active caspase, and this cellular process of apoptosis starts and cell will die. So generally when the cell is functioning, this will be inside the mitochondria, it will not come into the cytoplasm. Why it will not come into the cytoplasm? There are many factors that will keep it inside. So I'll just give you a rough idea about that factors. So there will be balance between certain genes which will control this process of apoptosis, some pro-apoptotic genes and anti-apoptotic genes. Okay, pro-apoptotic genes generally in a normal functioning cell. So pro-apoptotic genes and anti-apoptotic genes or anti-apoptotic factors. So there will be balance between these two, and these things will be functioning normally. But what happens, I'll just tell you. Just remember this anti-apoptotic factors or anti-apoptotic genes. So pro-apoptotic genes are examples are BAK, BAX. Anti-apoptotic genes examples are BCL2 and BCLX2. Okay, so these are the classical examples of this anti-apoptotic factors and pro-apoptotic factors. There is normally balance. Okay, so normally there will be certain tissue factors and tissue growth factors that will stimulate the cell, and normally this anti-apoptotic genes will be functioning. And these anti-apoptotic genes generally, suppose these are the pores of the on the mitochondrial membrane through which cytochrome C can enter the cytoplasm. So these pores are kept blocked by this anti-apoptotic genes or factors like BCL2 and BCLXL2. So they will block these things. Okay, they will keep this blocked so that cytochrome C does not enter into the cytoplasm from this mitochondria, right? Whereas this pro-apoptotic genes, they will cause the cytochrome C to come inside the cytoplasm. Okay, so generally they are kept under control, but whenever there is a withdrawal of the growth factors or any other things which leads to the starting or initiation or irradiation or cell damage, so these growth factors are not there, then these intrinsic pathways are activated, and this cytochrome C, these genes are activated, that is this BAK and BAX genes, and they causes the permeability of this mitochondrial membrane, and cytochrome C will now enter into this cytoplasm. Okay, when there is an imbalance between anti-apoptotic and pro-apoptotic factors. Okay, remember this anti-apoptotic and pro-apoptotic factors. So when there is an imbalance and when this cytochrome C enters the cytoplasm, okay, from the mitochondria, now this will bind with one more chemical known as apoptotic activating factor 1. Cytochrome C in the cytoplasm now will bind with one more in the cytoplasm, apoptotic activating factor 1. When these structures are combined together, when they bind together, then they will activate the inactive pro-caspase that is inactive to the active form, that is the caspase 9, from the inactive to active form, that is which was inactive. Now this caspase is active, and which it will activate? Caspase 9. There are so many, around 20 caspases are known. So the caspases 9 molecule or a protease molecules are activated. Now these caspases, as I said you, it is mainly of two types. One is initiator caspases, another one is executioner caspases. So initiator caspases are the one which gets activated initially, whereas executioner caspases are the ultimate caspases which will destroy the cell and lead to the process of apoptosis. So first, this caspase 9 is an example of an initiator caspase via intrinsic or mitochondrial pathway. Okay, So Caspases 9s are activated because when cytochrome C combines with the apoptotic activating factor 1, so they will convert pro-caspase 9, which was inactive, to the active caspase 9. Now this active caspase 9 or this enzyme or this is a protease, now it will convert the or it will stimulate the executioner caspases. So executioner caspases are the final caspases which will digest it will act as an endonuclease, it will digest the nuclear material, cytoskeleton and the other cellular organelles. So the executioner caspases, the name given are executioner caspases are 3 or 6, executioner caspases 6, these are stimulated, these will be inactive pro-caspases, and when this is activated it will activate executioner caspases 3 and 6, and this ultimately this will digest this nuclear material. Okay, so this is the story of the intrinsic pathway, that is the mitochondrial pathway. Now the process of apoptosis also occurs through the extrinsic pathway or the death receptor pathway. What is the mechanism? I hope this is clear. Okay, intrinsic pathway, those who didn't follow, just please rewind this video and please check out once again, the concept will be more clear. Okay, now through the extrinsic pathway, that is the death receptor pathway. So on the cell membrane generally there will be certain receptors are present. These receptors are known as death receptors. There is other name given to this receptor, FAS receptor, also known as CD95 receptor. So this receptor basically they are from the family of TNF, that is tumor necrosing factor 1. Okay, they belongs to this family and the name is FAS receptor or death receptor or CD95 receptor. Okay, so generally this is extrinsic pathway, they are stimulated by some extrinsic factors. Intrinsic factors, that is from the mitochondria, so from the cytotoxic T cells. So some cytotoxic T cells will stimulate this cells for the process of apoptosis. How? So they have one on the cytotoxic T cell, they have a ligand known as FAS ligand. Okay, so when this ligand binds with this FAS receptors, then whole story or mechanism of activation of caspases starts here. So this, when this FAS ligand binds with this FAS or CD95 receptor, then what happen is this, the lower portion, that is in the cytoplasmic portion, this is known as death domain of the FAS receptor. Okay, basically these are protein in nature. Whenever they are binded, this ligand from this cell, that is this T cell binds, then what happen is again 2 or 3 FAS receptor will be joining together, they will be binding together and the death domains are activated, and these death domains they will bind to one more protein, they are known as adapter proteins. Okay, and this adapter protein also has a death domain. Okay, and this death domain of the adapter protein is known as FADD, that is FAS associated death domain. Okay, these adapter proteins also has a death domain. So when they bind to this FAS molecules, so this is known as FAS associated death domain. So this FAS associated death domain, now what it will causes is, it will causes the activation of the initiator caspases through the extrinsic pathway, that is caspase 8, and in human being caspase 10. Okay, caspase 10. Remember, by the intrinsic pathway the caspase 9 was activated, whereas from the extrinsic pathway caspase 8, and in human caspase 10 are activated. That is, it was in pro-caspase 8 or pro-caspase 10 which gets activated to caspase 8 and 10, and after that activation remaining story is same, that is they will stimulate the formation of this executioner caspases, that is caspases 3 and 6, that are same, the same story happens, and ultimately this apoptotic bodies are formed. Okay, so both by the intrinsic and extrinsic pathway ultimately the cellular damage takes place, and the cell membrane, there is a shrinkage of the, what are the morphological changes of this by the intrinsic and extrinsic pathway. So ultimately through this caspases, executioner caspases, cell membranes gets shrinkage of the cell and formation of the apoptotic bodies takes place. So morphological changes, shrinkage of the cell, formation of the apoptotic bodies, condensation of the nuclear chromatin, And one more thing is on the cell membrane. On the cell membrane, this is basically cell membrane is a lipid bilayer membrane. Okay, so one lipid known as phosphatidylserine. Okay, phosphatidylserine. So this is present usually on the inner aspect of the cell membrane. Okay, so during this process of apoptosis, what happens is this is flipped to the outer aspect of the cell membrane, and phosphatidylserine will be now present on the outer aspect of the cell membrane due to the process of apoptosis. So these apoptotic bodies which are formed, so if you remember, apoptotic bodies basically a membrane bonded bodies which has the degraded cell organelles and the nuclear material. So this ultimately, these apoptotic bodies are engulfed by macrophages. Okay, macrophages will digest this. Okay, so the chemicals which will attract this are known as opsonins, and ultimately these apoptotic bodies are digested by the macrophages, and there will be no remnants of the cell seen, and surrounding there will be no inflammation as compared to the necrosis. Okay, so this is all about the apoptosis. So we'll discuss about the other things in the next video. Thank you very much.